a picture for going live. That was really hilarious. I'm trying to get ready this evening, and it snapped a photo before I even knew what was going to happen. So here I am live, Bio 123. You guys have a test on Wednesday over chapters 6, 7, 8, or 7, 8, 9, which is cellular respiration and cell cycle and meiosis and sexual reproduction and inheritance. So if anybody has a question, now's the time. Let's go for it. When we can address all your potential questions here. I don't have an easel yet, but I've got my trusty whiteboard here, and we're ready to go. So if anybody's ready about any of this stuff, let's see what you got coming in with some questions. I know I've had a few that, oh, my dog is in the house. Yeah, my wife's off, and the dog is here, and he wants to play fetch with me. Okay, so I can tell you in chapter 7, which is about cellular respiration, that's a really complicated subject. And one of the things that is going to be really important that you understand is this idea that cellular respiration is about taking energy that's stored in the foods we eat. So you go eat a pizza, right? And it's got a crust, which has lots of carbohydrates, it's got some cheese, which has some fats, and it's got some maybe some pepperoni on it, or if you're like me, I always get bacon on my pizza, and it's got lots of potential energy in it. But we can't really use that energy in those bonds directly. So what cellular respiration does is cellular respiration breaks down all of these chemicals, all of these carbohydrates, all of these fats and proteins. It breaks them down, so it oxidizes them, and it transfers those molecules, the energy in those molecules, to ATP. And of course, ATP is the energy currency of life. And that's the source of energy. That's the source of chemical energy that our cells rely upon. So if you're eating a pizza, you take the energy in the carbohydrates and you break down that carbohydrate through 4 steps: glycolysis, pyruvate oxidation in the Krebs cycle, and then of course the secret to life, which is oxidative phosphorylation, and you transfer that energy to ATP, which can then be used by your cells to do all kinds of different things. And I can tell you, if you've ever been in my class for very long, one of the things I talk about a lot is that the most important step in cellular respiration is understanding oxidative phosphorylation. That's the 4th and final stage of cellular respiration. So the first 3, what you're doing is you're taking an organic molecule, like a carbohydrate, and you're going to just oxidize it all the way to carbon dioxide. Every carbon, you're going to have those electrons and hydrogens just ripped off of them. That's the energy. We're going to take that energy and we're going to rip it away from the carbons. And by the end of the Krebs cycle, also known as the citric acid cycle, by the end of that carbon is just going to exit the cell as carbon dioxide. But we've managed to transfer that energy to something called the electron carrier. It's called NADH. And during the Krebs cycle, we also transfer those electrons and hydrogens to FADH2. And by the end of the Krebs cycle, we have 10 of these and 2 of these. And the magic, the secret to life is this thing called oxidative phosphorylation, right? So oxidative phosphorylation is the final part of cellular respiration, and it's where you make the vast majority of your ATP. I mean, glycolysis only makes 2 ATPs through a process called substrate-level phosphorylation. And I can come back to that in a few minutes and talk about substrate-level phosphorylation, but it's not very efficient. And Krebs cycle glycolysis, glycolysis makes 2, Krebs cycle makes 2 ATPs through substrate-level phosphorylation. And when we get to oxidative phosphorylation, we make like 32 ATPs. And it depends. It's not an exact number. It could be, between 28 and 32. So I always say oxidative phosphorylation is like one of the most important things for cellular respiration in terms of like this is where the magic happens. And let me explain to you really quick this thing about oxidative phosphorylation, because it's just so important. Okay, we're going to erase this. Now, to understand how oxidative phosphorylation works, we need to know a little bit about the mitochondria. Now, for those of you that have seen me before, you know that the mitochondria is like my favorite organelle. I know it's weird to have a favorite organelle, but I do. And it's a mitochondria because the mitochondria make everything possible. And my dog is trying to play fetch with me. He keeps putting a ball in my lap. Okay, so here's a mitochondria, commonly known as the powerhouse of the cell. And if you've watched my other videos, you know that it's actually a little bit more than just the powerhouse of the cell. But we have these two membranes. We got an outer membrane, and over here we have the inner membrane. This space in here is called the intermembrane space. And you know, if you're like my age, and every time you hear the word membrane, you can't help but think insane in the membrane. Cypress Hill, man, you stuck with me my entire life. Insane in the membrane. But we had this outer membrane, we've got this inner membrane, we have the intermembrane space, and then here we have the matrix. So this is the shape of a mitochondria. Now, understanding the shape of this mitochondria is really important for understanding how this process of oxidative phosphorylation works, right? Like this is like the key to so many things. This is where we make most of our ATP. I mean, this is where eukaryotic cells become possible, right? I mean, this is where like multicellular life becomes possible. It's through a process called oxidative phosphorylation. And it's split into two different things. One is called the electron transport chain, and we also have this other second part called chemiosmosis. And anybody that sat through one of my classes knows that chemiosmosis, I refer to this constantly as the secret to life. Now let's talk about this. So we've gone through cellular respiration. You know, we've got glycolysis, pyruvate oxidation, Krebs cycle. Those three stages of cellular respiration, they rip the electrons away from your organic molecules. We usually talk about glucose, but it could be a protein, it could be a fat as well. We rip those electrons away. Those electrons have energy, and we rip those hydrogens away. And we've transferred that energy to these 10 NADHs and these 2 FADHs as well. These molecules, that's what's carrying your energy from your glucose molecule in cellular respiration. Okay, so how does this whole thing of oxidative phosphorylation work? Well, first of all, the name oxidative tells you something. We're oxidizing something. If something's being oxidized, something's losing an electron. If something's being oxidized, something has to be reduced. That's why we call them redox reactions, because they always occur together. Oh, great handwriting there. I failed at handwriting. So oxidative phosphorylation, we're going to oxidize something. Electron carriers. Could they be carrying some electrons that we might oxidize? And we call them redox reactions because if something's being oxidized, if we're going to lose electrons here, somebody's going to gain an electron. Phosphorylation. That tells me I'm going to stick a phosphate group on something. And then we also know that oxidative phosphorylation occurs through two steps: the electron transport chain and chemiosmosis. Okay, so let's go into this. Let's get in here. We got this inner membrane of the mitochondria. And on this inner membrane, we have 4 large protein complexes that we call the electron transport chain. Now, complex 2 isn't as big as the other ones, but we got complex 1, complex 2, complex 3, complex 4. They all have their own names. Don't worry about that for right now. We can come back to that later. So let's zoom in to this area of the mitochondria right here. I'm going to draw another large protein complex called ATP synthase. So we've got complex 1, 2, 3, and 4, the electron transport chain. Electron transport chain. And I draw ATP synthase. That's something to do with chemiosmosis. So what I'm going to do, we're gonna zoom into here, and I'm going to explain to you the secret to multicellular life. Like, this is why animals exist. It's pretty crazy. Okay, so we're on the inner membrane of the mitochondria here, and I've got complex 1, complex 2, complex 3, complex 4, and I got another complex over here. 1, 2, 3, and 4, and this complex is called ATP synthase. Now we're going to come back to ATP synthase, but here's how this works. Okay, oxidative phosphorylation. I've got these electron carriers, NADH. This thing has been reduced. It's carrying the electrons from the organic molecules we've had. Boom, it's going to get oxidized to NAD+. We're going to rip these electrons and hydrogen off of it, and we're going to take that proton, which is a hydrogen ion, we're going to pump it into here, into the intermembrane space. Complex 2 is going to oxidize FADH2 back to FAD, and this will also be used to continue the Krebs cycle over and over. Now, here's my electron transport chain. I'm pulling these electrons off of here. Think electrons, energy, electricity. This does active transport. Complex 1 oxidizes NADH, oxidative phosphorylation, to NAD, and it's going to use that energy of the electron to do active transport and pump a proton into the intermembrane space. Complex 2 is going to oxidize FADH2 to FAD. Now it has high energy electrons. Complex 3 is going to serve as a proton pump. It is going to do active transport and it's going to pump protons into the intermembrane space. And complex 4, it is also going to pump protons into the intermembrane space. So the energy to drive this active transport is coming from the electrons that we snagged from our organic molecules. So when you drink some sugar, right, that sugar has energy. Well, we break down that sugar, we pull those electrons off of it, the electrons drive the electron transport chain. The electron transport chain oxidizes our electron carriers, NADH, FADH2, and uses the energy in these electrons to pump protons into the intermembrane space. And what we create is what we call an electrochemical gradient. Electrochemical, this is telling you something. We get an electrical gradient. You ever seen a battery, like a 12-volt battery from your car, and it's measuring 12 volts? That 12 volts is the difference between the positive and the negative end. So as I pump these protons in here, protons are positively charged. We build up a positive charge on the intermembrane space relative to the matrix of the mitochondria. So just down here, draw our mitochondria really quick. So we're pumping the protons in here, like this, and we get a positive charge over here. That's the electrical part, but because I also have more protons over here, that's the chemical part of this equation. So hence I have both an electrical chemical gradient. This system is now out of equilibrium. These protons don't like being out of equilibrium. They want to come back into the matrix and reach equilibrium with the protons in there, but they can't. The reason why they can't is because the inner membrane, like all cellular membranes, are made up of phospholipid bilayer. And we know that ions just don't really travel through a membrane very well. So now we have this thing called ATP synthase. ATP synthase is a large molecular complex. And what's going to happen is it's going to facilitate the diffusion of these protons down their concentration gradient. And what they're going to do is they're going to flow through this ATP synthase. Now, because all these hydrogen ions, protons are the same thing, they're going to flow through here. They're flowing through there in one direction. And the cell can harness that to do work. And the work they're going to do is the oxidative, this is the phosphorylation part. We're going to take adenosine diphosphate, plus a phosphate group. I draw the circle around the P so you know it's a phosphate group versus a phosphorus atom. And we're going to make ATP. This process of the protons flowing through ATP synthase to phosphorylate ADP to make ATP, this is chemiosmosis. In fact, osmosis means to push because these protons are pushing their way through. In fact, we call it the proton motive force. But for now, just go with, we've got the chemiosmosis. These protons are pushing their way through. This ATP synthase uses that kinetic energy to phosphorylate ADP to ATP. And this makes somewhere in the ballpark of 28 to 32 ATPs. And before this, we've only got like 4 ATPs from substrate-level phosphorylation. I mean, that's it. So that's pretty amazing that this process of chemiosmosis makes so much ATP. Now, earlier I said, chemiosmosis. I mean, this is the secret to life, right? Because every single cell on this planet makes ATP through chemiosmosis. Now, some of them use different ways of removing the electrons off the electron transport chain. So if you had no way to remove these electrons, then the electron transport chain would be reduced. Active transport would stop. You would have no more chemiosmosis. Now comes in oxygen. Oxygen is what we call this. Oxygen is called the final electron acceptor. Because here's what happens. This is brilliant. Oxygen is the second most electronegative element in the universe. I mean, this oxygen, what we see is O2. This molecule loves electrons. And if you remember covalent bonds, right, they're made by sharing electrons. But what oxygen does is it oxidizes everything around it. I mean, it oxidizes membranes, it oxidizes proteins, it can oxidize your DNA, and it can cause a lot of cellular damage. And in fact, part of our own aging process is caused by an accumulation of oxidative stress over time. So early on in life, about 2 billion years ago, there was a lot of O2 in the atmosphere from photosynthesis. This is, of course, a byproduct of photosynthesis. And it was causing problems for life on this planet because the oxygen was just oxidizing everything. It was killing stuff. But eventually, some bacteria figured out a way through evolution by natural selection to take complex 4. In fact, complex 4 is called Cytochrome Oxidase. And what complex 4 does, these electrons are going through the electron transport chain, and at every step they lose energy. They become more and more oxidized, which means it's harder to pull them up. But oxygen, being the second most electronegative element in the universe, picks up the oxygens from complex 4 as the final electron acceptor, and then it picks up the protons flowing through here, and it makes water. Wow. We took an incredibly reactive molecule. I know oxygen is an element because it's an element, but because it's an O2, it's a molecule. But we can use this cytochrome oxidase, this complex 4. It sticks the electrons on the oxygen, it picks up the hydrogens, and it forms water. And because oxygen is so electronegative, the electrons is like going down a ball, like a ball rolling down the stairs. They can go a long ways down in energy, which means we can extract a lot of work out of these electrons. That's amazing. And by getting so much work out of these electrons, you create a larger electrochemical gradient. We make ultimately more ATP. So by using oxygen as a final electron acceptor, we can now make way more ATP out of a single molecule of glucose. And I cannot overemphasize how amazing this is, because using oxygen as a final electron acceptor for aerobic respiration at the cellular level, I mean, this is why eukaryotic cells exist. This is why animals exist. This is why we can be a large multicellular organism and be like that. Thank you, oxygen. Okay, so I hope that helps out. I've got another question over here from Jordan. Can I go over Proto Oncogenes? All right. Okay. So let's gonna switch gears totally now. So good question, Jordan. Let's go over proto-oncogenes. So we got proto-oncogene. Now, before I go on to this, let me just say for a second that genes don't exist to cause disease. I mean, that's really important, right? Because that's like saying my liver exists to cause cirrhosis of the liver, or I've got lungs to cause lung cancer, or heart to cause heart disease. No, genes all serve a function in our bodies. Now, the problem is that when a gene becomes mutated or damaged, it may not function properly. So proto-oncogenes are involved with cancer. Now, if you remember, every cell in our body is under strict control, and we have what is called the cell cycle. We've got G1, S, G2, and M. This is the mitotic phase. This is where the cell actually divides. This is the first gap. This is synthesis, where you make your DNA, you copy it, and this is the second gap. And the gaps get their name from the gap in time between when a cell actually divides and when they divide their DNA, or replicate their DNA, I should say. So we have all these checks and balances in the cell cycle. We have a checkpoint right here called the G1 checkpoint. We have another checkpoint here called the G2 checkpoint, and we actually have something called the metaphase checkpoint. Now, what these checkpoints do is, okay, for any cell to commit to the cell cycle, it has to receive a signal, some type of growth factor. So it's got to produce a protein to produce that, to receive that signal. And then it's got to check its DNA. Hey, did I replicate my DNA okay? Do I have enough energy to reproduce? Can I repair my DNA? You get it. So there's all these checks on the cell cycle. So these proto-oncogenes are genes that are often involved with regulation of the cell cycle. Are you receiving the signal to divide? Are you checking your DNA for damage? Are you able to repair your DNA for damage? Okay, you get the idea. So that's what we call a proto-oncogene, are genes involved with regulation of the cell cycle and fixing and repairing DNA damage. Now, what happens with DNA? Every form of cancer is a result of damaged DNA, damaged genes somewhere. So proto-oncogenes are the genes that are often involved in the cell cycle, right? So what happens is, if you have a few mutations, these proto-oncogenes become oncogenes and they lead to cancer. So you can imagine, okay, I finished dividing my cell, I go into G1, I'm growing, I'm adding cytoplasm, I'm adding on to my organelles, I receive a signal to transition from G1 into S phase. I'm going to commit to the cell cycle by replicating my DNA. But before I do that, I want to check my DNA for damage and make sure there's nothing wrong with it. And if it is damaged, I'm going to come out of here, go into a gene not phase, right? So there's all kinds of genes involved with that. I'm either going to be able to repair my DNA, and if I can, great, I'm going to continue on to the cell cycle. If I cannot repair my DNA, I'm going to commit cell death. I'm going to commit suicide through a process called apoptosis. So an oncogene is a proto-oncogene that's involved with these problems, with the cell cycle. And when they become mutated and no longer function, now we've got an oncogene, a gene that leads to cancer. So, hey, I've got the signal to reproduce. I check my DNA, I've got damage. All of a sudden, I don't recognize the damage. I don't repair the damage. But we're just going to go ahead and reproduce our DNA anyways. That gene is not cancerous because it's leading to cancer. So now I'm going to commit to the cell cycle. I'm going to copy all of that damaged DNA. I'm going to divide it. Now both my daughter cells have that damaged DNA. Oh, wait. You know, every generation there's like 1,000 mutations. Doesn't matter where they're at. Oh, I don't care about these mutations. I don't need to repair it. So I'm going to go back on to S phase. And over time, what happens is on and on and on and on, the cell cycle goes, and we accumulate more and more damage. And that's how oncogenes work. Okay. Heavenly Sarah, do hermaphroditic organisms reproduce asexually or sexually? Man, that's a tough one. The answer is they actually reproduced sexually. Here's how a hermaphrodite works. So a hermaphrodite means you produce both male and female parts. So think of like a flower. A flower has pistils and they have stamens. The pistils are producing the ovaries and the eggs, and the stamens are producing the pollen, which is the male gametes. So if an animal is a hermaphrodite, like an earthworm, if you've ever been fishing, there's little earthworms. They're actually hermaphrodites. What they're doing is they're producing both the male and the female gametes, which means a hermaphrodite is neither male nor female. And a hermaphrodite, a lot of times in nature, can't self-fertilize, but sometimes they do. So even though it's the same organism producing an offspring, it's still sexual reproduction because they're producing male and female gametes. They're still combining two genomes into one. So it is a form of sexual reproduction. So Heaven, that's like a really good question. Like, nicely done on that one. So even though an organism is like producing its own offspring without like going through finding a mate, it's still actually sexual reproduction. Okay. Jared Fuller, I just want to say, howsome are you, and go Knowles. Thanks, Jared, for the call out on that one, and go Knowles. I'm wearing my Florida State t-shirt today because that's where I graduated from. Okay, Holly has a great question. Can you explain the difference between substrate-level phosphorylation and oxidative phosphorylation? Fantastic question. You're going to see this. Let's begin with what we call substrate-level phosphorylation. And let's break this words down so that we can understand them, because, man, that's substrate-level phosphorylation. Let's call it like it is. That's a mouthful. Anytime you see the words phosphorylation, what that means is you're going to add a phosphate group to something. And remember, a phosphate group is this functional group here. Okay? That's a phosphate group. And I'll write it like this so you don't get like phosphorus confused with the phosphate group. So phosphorylation means I'm going to add a phosphate group onto something. That's what phosphorylation always means. Now, substrate level. Okay. To facilitate chemical reactions, to make a chemical reaction speed up and go faster, we use enzymes. So let's take an enzyme. Let's say I've got an enzyme that looks something like this. Okay. And what I have here, these are my active sites. Okay, I've got an active site, and this is going to actually bind to my ADP. This will be my phosphate. And what this enzyme is going to do is stick this phosphate group onto this ADP to make ATP. The adenosine diphosphate, the ADP, and the phosphate, these are my substrates. So in substrate level phosphorylation, I'm going to phosphorylate ADP to ATP, which means I'm going to go from having 2 phosphates to having 3 phosphates on here, and I'm going to use an enzyme to do that. That's substrate level phosphorylation, because I'm creating an enzyme. This is my enzyme, substrate complex, and like I said, it will do this. Now, this, of course, is an endergonic reaction. This reaction requires an input of energy because these are the reactants and this is the product. So if I were to draw it, this is my change in energy. I use delta G for Gibbs free energy. The reactants are down here, the products are here. The products have more potential energy than the reactants. So this is an endergonic reaction. It requires an input of energy. And specifically, for all of you chemistry folks out there, at standard temperature and pressure, it requires 7.3 kilocalories a mole of energy to phosphorylate ADP to ATP. So what substrate level phosphorylation is going to do, it's going to combine an exergonic reaction, a reaction that releases energy, like the breakdown of glucose. So when you break down glucose, it's going to release energy, and it's going to use that energy to make this phosphorylation. Now, here's the thing about substrate level phosphorylation. If you have 7.2 kilocalories a mole of energy from your exergonic reaction, that's not going to give you any ATP. It's like I go to a Mountain Dew machine. I want a Mountain Dew. It's $1.50. You put in $1.49. You do not get 90% of a Mountain Dew. You get zero, and it takes your change. It takes all this energy. Okay, let's say you're really thirsty. You put in a $5 bill, but your Mountain Dew only costs $1.50. It gives you a Mountain Dew, but it also takes your $3.50. So substrate level phosphorylation is kind of it's not very precise. I mean, you got to line up your exergonic reactions very closely with your substrate level phosphorylation, whether you get no ATP or, well, you get your ATP, there's my Mountain Dew, but you wasted all your energy. So that's substrate level phosphorylation. I'm going to use an enzyme to phosphorylate ADP to ATP. Now, let's go on to oxidative phosphorylation. I've just talked about that a bit, but oxidative phosphorylation is the fourth and final step of cellular respiration, right? And like I said, that occurs on the inner membrane of the mitochondria. We've got our electron transport chain, complex 1, 2, 3, and 4. And what's going to happen, the electron, we'll call it the ETC transport chain, what that's going to do is I'm going to take all the energy from the breakdown of my organic molecules. So imagine every time you go and get change, you store in a piggy bank. So it's going to pump protons into the inner membrane space, right? So every time I break down, I go through a chemical reaction that releases energy, I'm going to store that energy as an electrochemical gradient. And then what I have is I have this molecule called ATP synthase. Now, I've created an electrochemical gradient. These protons, they want to flow back down into the matrix of the mitochondria, but they can't cross a membrane. So we have what's called facilitated diffusion. These things flow through the ATP synthase. This part is called chemiosmosis. And every time we get enough energy storage, 7.3 kilocalories a mole, we make ATP. So the beauty of oxidative phosphorylation is as I go through like 11 steps of glycolysis, 1 step of pyruvate oxidation, and 8 steps of Krebs cycle, I can take all the bits and pieces, all the change of these reactions and store them as my electrochemical gradient. And then through oxidative phosphorylation, which includes both creating the electrochemical gradient through the electron transport chain, and now I can make all of my ATP through chemiosmosis, which is these protons flowing through the ATP synthase. So it's like really, really, really efficient. And the difference is I don't have to couple an exergonic reaction specifically with an endergonic reaction and match that perfectly to make ATP. Here I can store my energy like in a piggy bank, which is just absolutely brilliant, right? To make all of my ATP, and that's why like it's so efficient, and that's basically like every single cell on this planet makes ATP through this process. Okay, next question. This guy Brixton from Jared Fuller would like to know why women are from Venus and men are from Mars. That's a pretty funny question, actually. In our world of equality, both men and women are very, very, very similar. When it comes to intelligence, there basically is no difference between men and women. In fact, women actually make incredibly good scientists and incredibly good mathematicians. And, it's been said that sometimes every now and then, like the guy that got all the credit is really because his wife was pushing him and actually came up with the ideas, and the guy got all the credit for it. But the difference is men and women are wired a little bit differently in our brains. Our brains are different, and we do perceive the world differently. And I think that those differences, we need to talk about those differences between the way we perceive things, especially when it comes to emotions and empathy, to put those out there. And not just go around saying we're exactly the same, because we're not the same. Men and women we perceive the world differently, and we know this. And I think the more we talk about those differences, and we get to know each other, then all of a sudden we have better communication, and there's not these missed in translation type of things. But when it comes to intelligence, like every data point has ever shown that we're really, really different, except men tend to have more testosterone. So yeah, that can make us a little bit more gung-ho about things. Women tend to be a little more empathetic, but those are general trends. And something that's very important is that those are trends only. There are guys that like don't really want to be aggressive, and there are women that are. So like I said, hey, when it comes to doing science, everybody is capable of doing science. I mean, what it really takes is be curious about your world, right? Like, go make observations and ask questions. And, if you want to be a scientist, you can. Okay. Let's see. Jen Hester, does the way your partner smells really have an influence on whether or not you choose them? Actually, it does. Wow, that's kind of far out there, but absolutely. A lot of women have been known to choose their partners based on the way they smell. And in fact, there's been some really solid scientific studies that have shown that women prefer the smell of their partner. Now, you're going, "Whoa, guys, we all smell." But it turns out that we all have an immune system. And we have an innate immune system, and we have an acquired immune system. But our immune systems between different types of people can be, well, a little bit different sometimes. And it's called Mhc compatibility, or major histocompatibility, MHC. And it turns out that when people say, "Oh man, my partner smells so good," it turns out that it has to do with our immune system, and we're smelling differences in pheromones between us. And when you find a partner with a very different immune system, they often smell really good to you, and you're attracted to them. So if you've ever had like this attraction towards somebody, you're like, "Whoa, I'm so attracted to you, and I don't know why." It could be this MHC that you're picking up on, or a pheromone that you're picking up on. And the idea is that if your partner has a very different immune system from you, then, well, your offspring will have the best of both worlds and will be basically more immune to different types of diseases in the long run. So yeah, it's a real thing, actually. Okay. Oh, Jen Hester again. Are GMO products really safe for us to eat? Absolutely. There is nothing wrong with eating GMOs. I mean, if I take a gene to make a tomato freeze resistant by putting a gene from a fish that doesn't freeze, it's not going to make it taste fishy. What you have is you've got a fish that produces a chemical that's actually really good for us to eat. We put that in a tomato or a strawberry. It doesn't make it taste like a fish. It makes that organism resistant to freezing. And in fact, in some ways, we actually get the benefits of that cold water fish now in a strawberry. So now that strawberry or that tomato, guess what? It's even better for us now. Okay, Holly Hall. Do we need to memorize what step of cellular respiration makes, for example, glycolysis, 2-ATP pyruvase, and 2-NADHs? Yes, absolutely. 4 stages to cellular respiration. Here's what we need to know. Oh, my poor kid in the background. She's having a rough time. Okay, we got glycolysis. Remember, glycolysis, glycolysis. We're going to split sugar. Glycolysis occurs in the cytoplasm. It occurs independently of the mitochondria. It occurs independently of whether or not you have oxygen present. But glycolysis, what it's going to do is it's going to split the sugar into 2 of these pyruvate molecules. And remember, the glucose molecule C6H12O6. Hold on one second. All right. So if we split this into 2 pyruvates, these are 3 carbon sugars, and pyruvate is going to have a carboxyl group at one end. It's going to have a carbonyl group in the middle, and it's going to have a methyl group here. And this is our pyruvate molecule. Actually, it's pyruvic acid for all you chemistry buffs. Now, because we've broken down glucose into two pyruvates and we've started to oxidize it, glycolysis is also going to transfer some of these electrons to two NADHs, and we're also going to produce two ATPs through substrate-level phosphorylation. So that's glycolysis. Okay, that's what we come out of that. So you absolutely have to know that. Now, the next step, so we'll just keep our running tab over here. We got glycolysis. The next step is what we call pyruvate oxidation. Now, pyruvate oxidation, if there's enough oxygen inside the cell, pyruvate will be pulled into the mitochondria, and we're going to take this molecule. Here's our carboxyl group, and then, oh, I can draw that H better. We got a carbonyl, we got a methyl group here. And in pyruvate oxidation, what we're going to do now is we're going to oxidize pyruvate. And you see this carboxyl group, we're going to oxidize that to CO2. Now that makes sense, right? Because oxidation is losing electrons. So I'm going to pull this electron off of here. I'm going to take and break this bond. I'm going to break this hydrogen off. I'm going to form CO2 for each pyruvate. So for each glucose molecule, we'll get 2 CO2. Now, once again, I've oxidized something, something must be reduced. So my electron carriers, I'm going to reduce 2 NADs to 2 NADHs. So for pyruvate oxidation, I'm going to pop off this carboxyl group, form 2 CO2s. Because I'm oxidizing this, 2 NADs get reduced to 2 NADHs. This step does not produce ATP. That's important. No ATP produced here. Now this is called acetyl aldehyde. And acetyl aldehyde is going to form acetyl coenzyme A. And I'm going to get 2 of these because I've split glucose into 2. So I'm going to get 2 of these acetyl aldehydes, or specifically acetyl-CoA. This is my acetyl. I'm going to attach this to coenzyme A. And then we're going to do Krebs cycle. So this carbon here, these are nonpolar covalent bonds. They carry a lot of potential energy. In the Krebs cycle, what we're going to do now is I'm going to oxidize these carbons. I'm going to strip these electrons off of them, and we're going to transfer that energy to ATP and NADH. So the Krebs cycle is going to occur in the matrix of the mitochondria, and I'm going to get 6 NADH, 2 FADH2. Once again, that's from the oxidation of this. And because I've got 2 carbon dioxides, 2 carbons, and 4 total, because I have 2 acetyl aldehydes, I will produce 4 CO2s, and I will also get 2 ATPs through substrate-level phosphorylation. And of course, these numbers in Krebs cycle are for each molecule of glucose I started with. So by the end of these first 3 stages of cellular respiration, I now have 10 NADHs, 2 FADH2. So I've taken my glucose molecule, I ripped off all the electrons, all the hydrogens, I've transferred them to these electron carriers, and I've also made a total of 4 ATPs through substrate-level phosphorylation. And then the 4th and final stage of cellular respiration is oxidative phosphorylation. And what I'm going to do here is I'm going to oxidize these molecules. These are my electron carriers, and I'm going to produce somewhere in the neighborhood of 28 to 32 ATPs. Now, a lot of people like to say it's a specific number of ATPs. And it's not an exact number. It's somewhere in here, 28 to 32. Now, substrate level phosphorylation. Oh, yeah, glycolysis. You will always get 2 ATPs through substrate level phosphorylation. Krebs cycle. You will always get 2 ATPs through substrate level phosphorylation. And for those of you that are into chemistry, you'll know that it's actually GTP and oxidative phosphorylation, or in Krebs cycle, but we'll just say ATP because it actually gets converted to ATP. Then oxidative phosphorylation produces the most amount. So to answer that question, yes, memorize those steps of cellular respiration and make sure you know how much comes out of each one. Okay. Jen, all right, on a totally different subject. if a mother can't breastfeed her baby, is that bad? Okay, you know what? Breastfeeding, as I discovered, you would think is a mammal. Mammals have been around for like, 100 million years, and in fact, maybe even going back 200 million years. And one of the defining characters of mammals is we have mammary glands that produce milk to feed our offspring. So we have this very extended parental care. So what's really interesting is that the milk of a mammal is tailored to its offspring. So cow milk is very different from whale milk, which is very different from bat milk, which is very different from rat milk, which is very different from goat milk, which is very different from human milk. But we all produce milk based on the needs of our offspring. And if you're a marsupial, the ratio of like proteins and fats and carbohydrates really changes a lot. And in us, not so much. So yes, we've produced this milk for our babies. As a human, our breast milk is completely tailored for the growth and needs of our babies. And not only is it geared toward growing them, but it also is how we build the immune system of our babies. Like, so the babies can actually get an acquired immune system from their mom. Now, that being said, something that I learned about humans is that, wow, getting your babies to breastfeed is really, 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 really hard. I don't know, understand how we like survived as a species, because watching how hard it is to get our children to breastfeed. I guess before modern medicine, we actually had a 50% die rate or death rate of our babies before the age of 5, before modern medicine. However, in our modern world, breastfeeding is always the best. If you can do it, go for it. It's absolutely the best way to feed your baby. However, if you can't, you should never feel ashamed. You should never feel like you failed your child, because sometimes it's too painful. Sometimes your baby won't latch on. It's amazing to me that sometimes babies don't latch on. In that case, the wonderful thing with modern medicine is, well, guess what? We have breast milk supplements. So you can actually go buy formula at the store. You can get breast milk from other moms. There are other ways to get breast milk. So you know what? If you can't get your baby to latch, you're not a failure in any way. There are other alternatives. And that's one of the things we can say thank you to modern society, because instead of watching your baby die of starvation because it won't latch, or you can't produce milk in time, in our world, we can actually get around that. But if you can breastfeed, anywhere between 6 months to a year, in fact, the World Health Organization recommends that you actually feed your baby for a year. And that's about it. I mean, if you want to go a little bit longer or a little bit less, that's okay. All right. Jordan Brewer. Does caffeine have an effect? Does caffeine have an effect on mitochondria, and why do we get energized from it? That's an amazing question. And that actually is a little bit beyond some of the things that I know. But I can tell you what we have is we have these receptors in our brain that affect our metabolism. So the rate at which we burn calories is called our base rate metabolism. And that's the rate at which we generate heat for our body. That's how we maintain all those sodium and potassium pumps, you name it. Now, as it goes into wintertime, our metabolism changes compared to wintertime. And as we go from a baby to an adult to an old person, our base rate metabolism changes. So what caffeine does is it actually ramps up our metabolism by sending signals to our cells going, you need to increase the rate at which you're burning calories. So yes, caffeine does affect that base rate metabolism. But to be honest, I don't actually know exactly how it affects the rate at which mitochondria burn calories or burn sugar. That's an amazingly good question. Awesome. I'm sorry, Jordan, that I can't answer your question better than that, but great question. And I'm gonna actually have to come back to that and go look up for the answer myself. All right, Holly Hall, can you explain the importance of fermentation? Okay, well, other than brewing good beer, wine, and then distilling that into alcohol for spirits, animals and plants and all kinds of things actually rely on fermentation to continue making ATP. Now, of course, in modern society, for any of you that want to go into brewing, wow, what an amazing time to become a brewer, because people are making billions of dollars off of that. But let me explain it to you. If you've ever sprinted, if you've ever bench pressed, and you're hitting 7, 8, 9 reps, hitting failure around the 10th rep, basically what's happened is you've run out of ATP to make your muscles contract. That's what failure is all about. Or let's say you're sprinting and you hit that 100 yards and you just can't go anymore. You've run out of ATP. You've gone what we call anaerobic, and you've gone what's called fermentation. And so this is what happens in our cells. We have what is called lactic acid fermentation. So, at the end of glycolysis, we produce something called pyruvate, which I just talked about earlier. And pyruvate looks like this. It's actually called pyruvic acid, but this is pyruvate. Now, glycolysis requires no oxygen. You don't have to have any oxygen, and it makes 2 ATPs. Okay, through substrate level phosphorylation. So when you go into fermentation, let's say you're bench pressing, you're sprinting, or let's say you are a yeast cell and there's no oxygen around, you got to have ATP. I mean, at least a little bit. So what lactic acid fermentation is going to do is it's going to reduce pyruvate to lactic acid. Now, the way it's going to do that is here's our carboxyl group, and then we're going to reduce this right here to a hydroxyl group, add another hydrogen in there, and here's our methyl group. And this molecule has now been reduced, but if something's been reduced, something has to be oxidized, and the purpose of this fermentation is to oxidize NADH back to NAD. And this is required for glycolysis to occur, right? So without a supply of NAD, glycolysis can't happen. And without glycolysis, you can't even make this. So the purpose of fermentation in our cells is, hey, let's say we've outstripped our oxygen demand, we can still produce a little ATP through lactic acid fermentation. So we take pyruvate, we reduce it to lactic acid, and the importance of that is to oxidize NADH to NAD. We're adding in a hydrogen, There we go. Oh, there's some water involved in this too, but we won't get into that. And now we have an oxidized form of NAD, and this can be used once again in glycolysis. Now, if you're a yeast and you're going to brew some beer or wine or mead, if you haven't been to my lectures, you know that alcohol fermentation, of course, was the start of civilization. So here's my pyruvate molecule. Let's talk about ethanol fermentation. So if you're a yeast and if you've ever brewed beer, well, they also do glycolysis. Like I said, almost every single cell on this planet does glycolysis. And for glycolysis to occur, you have to have a supply of NAD so that you can oxidize oxidize glucose and form NADH. If you're going through glycolysis, we form NADH, but to continue it, we need this. So we're going to reduce pyruvate, and this is ethanol fermentation. This is what happens when you make brew. This carboxyl group is going to become CO2. I'm going to oxidize that. But what I'm going to do is I'm going to make this molecule here. And if anybody starts to recognize this molecule, of course, this is ethanol. So our little yeast, baker's yeast, brewing yeast for beer or wine or mead, you can't let your wine or your beer, your mead ever get into contact with oxygen, because it will just go through the Krebs cycle and go through oxidative phosphorylation. But if you keep it anaerobic, here's our pyruvate at the end of glycolysis. We're going to oxidize NADH to NAD. At the same time, we're going to reduce, didn't spell that very well, we're going to reduce pyruvate, in this case to ethanol, and produce a couple of CO2s. So if you've ever had a beer, for all of you over 21, I know that you guys under the age of 21 are drinking warm milk or some Kool-Aid or something, but this is where the bubbles come from for beer. And if you've ever brewed wine, you have a little valve on there to release the CO2. All right, so that's the importance of fermentation. is that fermentation allows you to oxidize NADH back to NAD by reducing pyruvate so that you can continue with Krebs cycle, continue with glycolysis and keep making a couple ATPs. Okay, Jen, do vaccines cause autism? No, they don't. And in fact, man, that's, wow, yeah, there's no evidence. We've got 50 years, 60 years of people receiving vaccinations. And what we realize is that we know that there is no way that a vaccine can cause autism. No, we have learned a lot about autism, and we realize there's a strong genetic component to it, there's an environmental component to it, but in no way can a vaccine ever cause autism. There's no link, there's no causative agent. The problem is that when you start getting all your rounds of vaccinations, people go, oh, well, you know, my child started showing signs of mental impairment after that, or cognitive impairment. You know what? No way. It's a complete correlation. There's nothing in a vaccine that can make you autistic. It'd be like saying, I caught the cold and that made me autistic. It's about the same, right? So the way a vaccine works is you have what's called an acquired immune system. It's like Nietzsche. That which does not kill you makes you stronger. So everything you've ever been exposed to in your life, your body remembers it. So if you get exposed to it again, your body goes, I know you, I see you. And it just launches a full-scale attack and just crushes any type of pathogen before you even become symptomatic, at least most of the time. So there's no way that like getting a vaccine that has like a, you know, a little bit of an attenuated virus of like a measles, mumps, rubella, pertussis, whooping cough, tetanus, any of these things could affect your mental or cognitive state. It just doesn't work like that. That's like saying, you know, oh, I caught the cold and that antigen from the cold caused an antibody to be released, which caused mental impairment. No way. There's no way it causes brain damage. So, yeah, sorry. I know that all the anti-vaxxers talk about these conspiracy theories, but they're basically full of crap. Okay, Jen Hester again. Do you know why people draw stars like a star and the stars actually resemble the shape at all? You know, that's a pretty interesting question. What happens is when all stars are basically suns like ourselves. So every single star in the world, in the universe, and is circular. Oh, that's my, they're way more circular than that. I can't draw a very good circle. Maybe I could trace out my cup here and draw a circle. But all stars are totally spherical. And that's because of gravity. Now what happens is the way that we perceive light on our lenses or lights coming through our solar system causes the light to flare, and hence we see the flaring action of the light coming into our cameras, into our eyes, and that's what causes the star shape. And in fact, most of the time you see something like this, which looks more like the Zuni sun symbol out here in New Mexico. Okay, so Jen Hester again. All right, we're Ask a Scientist here. What is on a moth wing that falls off when we catch them and try to put them back outside when they end up inside the house? You know, have you ever caught a moth, right, or a butterfly, and you're like, oh, this poor animal's in my house, and I'm gonna catch it and take it back outside, and you get this little stuff on your finger, this little bit of, I don't know, dusting of stuff on your finger, and you look at your poor moth, and it's no longer as vibrant as what it was. Well, that's a very good question, and it turns out that moths and butterflies belong to a group of insects called I can't spell without my spell check. Lepidopterans. All right. Embarrassing. Lepidopterans. All right, embarrassing. I can't spell without my spell check today. But basically, pteran means wing. Lepido means scaled. So moths and butterflies actually have tiny little scales on their wings. So when you catch a moth or a butterfly in your hand, those little scales come off, right? And so all of a sudden they look a lot more pale, and you have little bits of their scales on your wings. So that's why moths and butterflies, you know, that's why they They lose stuff. There's this stuff that falls off. It's their little bitty scales. Hence the name Lepidopterans. Okay, Holly. In class, we talked about incomplete dominance. Can you cover that again? My notes say that the heterozygous have a phenotype similar to both alleles. Isn't Lepidopterans, isn't phenotypes the outward appearance? Okay, that's a really good question, Holly. Let me clarify a couple terms for you really quick. And I think this will really help out. Okay, first of all, we have what is called genotype, and we have what is called the phenotype. So the genotype is your actual genes. This is what your genetic makeup is. So let's talk about let's do something like a red rose, because you've probably seen white roses, red roses, and pink roses. And we'll use capital R for the red, and we'll use lowercase r for white. So we've got white roses and red roses. Now your phenotype is your outward appearance. What color is your skin? What color are your eyes? What color is your hair? Do you have curly hair, straight hair? If you're a flower, are you a red rose, a white rose, pink rose? Are you a purple flower, white flower, yellow seed? You get the idea. The phenotype is your outward appearance. And your outward appearance is based on your genotype. And don't forget your environment. And we're going to learn that tomorrow in class. That like skin color is totally a function of genotype by environment interaction. But for now, let's talk about incomplete dominance. Let's talk about roses. Red roses, white roses. If I cross a true breeding red rose with a true breeding white rose, every one of these has the variation in a gene that's for red, and we call that an allele. And this is another version, it's a white allele. Okay. Now, what we have here, big R, little r, we got the red allele and we have the white allele. And the term heterozygous means other, and heterozygous means you have two different alleles for a particular gene. So our gene here is flower color. Our alleles are red and white, okay? So if you're heterozygous, now that means you've got the red allele and the white allele. Now for incomplete dominance, here's what this means. In incomplete dominance, the phenotype looks blended. Now we're not blending our genes. I mean, these things are still discrete, but the phenotype looks blended. So when I get red and white, I get a pink rose. Because to have a red rose, you have to be homozygous. Homozygous means I carry two alleles for the same one, right? Okay. I got two of the same alleles. So now I've got homozygous, that makes me red. This makes me white. But if I'm going to be incomplete dominant, then I'm heterozygous, I have a red allele and a white allele. And because you only produce half the amount of the red, that means now all of a sudden I am pink. I'm neither red nor white. That's incomplete dominance. So the other one is hypercholesterolemia. That's also incomplete dominance as well. Okay, let's see here. Jen Hester. All right, once again, why do people get so worked up about the Rio Grande silvery minnow? Is this really a species worth all the money and effort to save? The answer is absolutely. You know, the Rio Grande River is the third largest river in the United States. I mean, right behind the Mississippi. And there's a lot of fish in the river. Believe it or not, there's actually fish in the Rio Grande. And whenever we start losing species to extinction, what that means is that ecosystem is becoming damaged. We're causing great harm to that ecosystem, and the species that were there can no longer live there, and they go extinct. So when we look at something like the Rio Grande silvery minnow, and we're like, wow, you know, this species is going extinct, we're spending all this money on it. Should we be spending money on saving this one fish? And the answer is absolutely, because we're not just trying to save this one fish, we're trying to save the entire Rio Grande ecosystem. So for instance, by saving the Rio Grande silvery minnow, you know, we actually have to reintroduce flooding into the river. Now that may not seem all that great to introduce flooding, but like the bosque, the cottonwoods along the bosque depend on flooding on the river. So do the silvery minnows. So if the silvery minnows go extinct, that means the ecosystem is no longer functioning correctly, it also means that we could lose our bosque. And by saving the silvery minnow, not only do we save all the other fish in the river, but we save our bosque, this beautiful ribbon of large cottonwoods that grow right along the stream. And if we lose that, it's just desert all the way down, and we lose a lot of the diversity in the whole region. So it's absolutely important to save the Rio Grande silvery minnow. Okay, Holly. Yep, I'll see you in class tomorrow too. Jen must be a fish biologist. Do I have a favorite fish species in the Rio Grande? Do I have a favorite fish? I would say long-nosed dace, Rhinichthyes cataractae. I also really like river carp suckers. Yeah, those are beautiful fish. And those are cool. All right. Oh, another one. How do I feel about introduction of the tamarisk beetle? So if you're not aware of it, in the West, we have this invasive species called tamarisk, also known as salt cedar. And this is a discussion for another time, but I'll briefly talk about it here. But we have all these invasive species in our country, and invasive species cause a lot of ecological damage. They drive species to extinction. They change the ways that ecosystems function, and they cost us literally billions and billions of dollars. So in the Southwest, one thing we deal with is a salt cedar, and it goes into our bosque and our other wetland areas and takes over. And there's been this really big push to remove it so they can restore native vegetation. So a few years ago, they released a beetle that actually kills the salt cedar. And well, you know, like, yeah, cool, it's killing off this invasive species. We can restore the native habitats and restore our native diversity. But it's really complicated. You see, whenever an invasive species comes into our environment, what it does is it creates new interactions with all the native species. And the native species actually become dependent on these invasive species. All right, so if you go remove the invasive species, what are the dependent species of that going to do? What's going to be the natives? If you don't immediately restore habitat, you further impacted those native species. And even worse, out here in the West, you know, we've been in a long-term drought. It's a thermal drought, meaning that our drought is caused mostly by a rise in temperature, not loss in precipitation. Well, that's lowered water tables. So now salt cedar can survive in areas where our native vegetation can't anymore. So now we go and remove all this salt cedar, which provided habitat for our native species, but the native plants can no longer grow there anymore. So we remove it, this beetle is wiping out all of these tamarisk, but the native plants can't grow back. So the reality is that the tamarisk beetle is actually probably causing more harm than good. But from, you know, that's my hypothesis. And that hypothesis needs to go out and be tested with actual data. And if the data comes back, whoa, the native species are blossoming with the loss of tamarisk, then I'm wrong. If the data come back and say, hey, yeah, we've got, we remove the tamarisk, we lose native species, then I would say, hey, we should not go so far to try to remove the salt cedar. In that case, the beetle will be bad. But it's one of those cases where we need data. Okay. Hey, at 8:38, been on for an hour and 10 minutes. It was a lot of fun tonight. I will see some of you in class on Monday. And for the rest of you, sign in next week on Sunday, and we will do Ask a Scientist again. Thank you, some people, for asking, like Jared and Jen, for asking some really cool questions not related to class. I love answering everything. So I will see you next week, same time, same place. Ask a Scientist.